All right, we're going to recap from last video where we went through meiosis all the way up to metaphase 2. So this is our first cell in meiosis. It's our interphase. I can recognize it due to the fact that it has this chromatin. We've got blue to represent chromosomes that were originated from the father of this cell, and the pink or red to represent the maternal contribution. We have two centrioles because they already replicated during the S phase. They progress into prophase 1, and in prophase 1 you see tetrads form, where tetrads are replicated chromosomes that are homologous to one another. So they have the same genes on them, but they have a different variation of that gene, and they pair up side by side. And what this picture shows is this crossing over. It adds variation to the outcome that would eventually happen through this process. So right now we have two tetrads formed, a nuclear membrane breaking down, and the centrioles migrating to the pole. So that's an indication of prophase 1. Metaphase 1 is going to look like this. You have centrioles completely at the poles, and we have our tetrads aligned at the equator. Now, the organization of the tetrads is arbitrary. That's the whole idea of independent assortment. These tetrads can line up in any fashion. There's only two tetrads here, but the more tetrads you have, the more variation you can get, and they can actually flip-flop. The paternal contribution, or blue, can be on top, or the pink could have been on top in the two variations that I did show in the last video. So with that, anaphase 1, the spindle fibers, which were attached to these replicated chromosomes, will retract, pulling apart the homologous replicated chromosomes from one another, or basically splitting the tetrad in half. From there, we have telophase 1, which kind of bypassed because I abbreviated this picture. But telophase 1, this membrane would have been one continuous membrane. The nuclear membrane would have been reappearing, but there would have only been one centriole. In the interim between telophase 1 and what this picture shows is prophase 2, the centrioles had to replicate. That was the only thing that replicated in between. The nuclear membrane is going to disappear, and inside of the nucleus are two replicated chromosomes, all right, one of each size. Depending on how they assorted independently back in metaphase 1, there is a possibility that these nuclei could have a different color chromosome in them. For instance, if the tetrads lined up opposite and maybe one of them flipped around, you might have a nucleus here on top that had the large blue and a small pink. That is still a possible outcome. That's the whole idea of variation because of independent assortment. Then we take a look at metaphase 2. So metaphase 2, the nuclear membranes completely disappeared, and the two replicated chromosomes line themselves up at the equator. So in both versions. So from here, I will show just side by side, and I don't have to just flip to the next thing. We have anaphase 2. So let's look at what would happen here. The spindle fibers would retract. The sister chromatids that make up the replicated chromosomes would pull apart. So the larger sister chromatids would be there, the smaller. Now they're called non-replicated chromosomes. So this is one non-replicated chromosome. There's another non-replicated chromosome. Down here, the other cell, which was made at the end of meiosis 1, is now going to pull apart these chromosomes. The large pink non-replicated chromosome, small and small. All right? So that's anaphase 2. The sister chromatids are pulled apart. In anaphase 1, replicated chromosomes were pulled apart. It's very important to know that the terminology, because that's going to be a good descriptor to explain what's happening at the different phases. After anaphase 2, we're going to go into telophase 2. So what that will look like is the top cell is going to have another cleavage furrow occur. A nucleus is going to reappear. Each cell that's developing will have its own centriole. The spindle fibers disappear. You're going to have a large and a small, large and a small. And then the same thing was occurring with this. Cleavage furrow present, nuclear membrane, centriole, but in this one, it was the combination of large and small of the pink variety. All right, So telophase 2 is somewhat happening once again at the same time cytokinesis is occurring. That's the whole cleavage furrow. So at this point, we'll show what happens at the end of this process. So you have so cytokinesis has completely occurred. We now have 4 gametes that were produced. Each one, nucleus reappeared completely. Each one only has 1 centriole. And now the contents now it looks like we've only made 2 variations of gametes. These 2 look identical in terms of their DNA, and these 2 at the bottom look identical. But what has actually happened, if you recall back in prophase 1, remember that sister chromatids of homologous chromosomes swapped information with the crossing over. So what would have actually happened is this one was the one that if we kept it up, we would have had a small piece of pink on the end, and this one we would have had a small piece of blue. So now we don't have 2 pairs that are identical. We now have 4 unique gametes, and that's what adds the diversity if we kept up the whole concept of crossing over throughout this entire process. So these could be potentially 4 sperm, or it could be potentially 4 eggs. So that's kind of the general overview of meiosis and the steps to get through from interphase all the way to telophase 2/cytokinesis. Okay?